Thomas, it's great to have you here at SciLife Lab, and congratulations to the prize. It's been such an honor for you.
Thank you very much.
So we have sources of DNA damage either from the outside or from the inside, spontaneous lesions. And I want to hear your opinion from working in this area for a long time. What the balance is it of course diet is important, but how important do you think diet is as compared to the spontaneous lesions?
Not very important at all, huh?
So, yes.
During the centuries, we have figured out there are certain mushrooms that are very poisonous and perhaps other food stuff that are not good for us, and they are not used in a normal Western diet anymore. Huh? So I don't worry about eating special kinds of food. Anything you can go and buy in a supermarket is fine.
People age, and no matter what you eat, you have a very similar lifespan. And we can see that individuals that have unhealthy diet, they still can be living for a very long time. They have a higher risk, of course, of getting cancer, higher risk of cardiovascular disease and other diseases. But the lifespan is very similar. And also looking at individuals, I'm amazed that we can see the difference between a 30 and a 40-year-old, or even a 60 or a 70-year-old. You can just look at the individual, and you can classify where they are. And this must be spontaneous damage, and not what you eat. So, to my mind, spontaneous damage is probably the most important when it comes to aging.
Yes, I agree with that. And it's some kind of myth that we are living in a big sea of mutagens, and that the food we are eating contains poisonous components or dangerous components. I don't quite know where this worry comes from, because it's not true. There are some things that are bad, and they have already been identified, like chain smoking cigarettes is not a good idea. But I don't think there is anything more to be discovered along those lines. There is nothing as nasty as inhaling cigarette smoke in the diets we have today.
I somehow agree with you with the conception of aging, that it is important. The most important is probably the spontaneous lesions. But when it comes to cancer, I also want to challenge you a little bit, because looking at different populations in different countries, we can see that in Sweden we have prostate and breast cancer being very common cancers, while in East Asia we have stomach cancers being very prominent. And it seems like we have a very lot of different cancers depending on where on the planet we live.
I think you are right. If a population like some areas of Asia have an increased level of stomach cancer, then I think it's just common sense to suspect that this has something to do with what you are consuming. And you should look carefully what you are eating. And we don't quite know what it is. It might be some of the pickled food that's popular mainstay of some Asian diets.
And also the Helicobacter pylori, is the bacteria that can cause the different ulcers, and they are also important when it comes to inflammation and causing cancer.
No, Helicobacter is a nasty thing, and one should be careful with exposure.
And I think so there is this environmental component and also the spontaneous component. And so this is a balance. And then when it comes to the spontaneous lesions, then which ones are the most important? In your studies, which ones were you thinking were the most important?
I think we have probably identify the most important ones, because they are the ones that are the easiest to find, the ones that are the most often.
Yes.
But there could still be
So
some
which
surprises.
are the ones that are the most frequent, just to enlighten the audience?
Well, you can lose genetic information, like a base falling off DNA, and that's a typical water-induced damage. We have large proportion of our cells are composed of water. There's nothing we can do that. We can't protect ourselves from the water in our own cells. We just, if there are rare side effects of that, we just have to live with that. And the way we live with it is to repair the
It's also
DNA.
how we use, because there is also a different rate of loss of the stability depending on how you're packing the DNA and how much you're using the DNA. So much of the recent studies have suggested that there is a lot of interference with replication and transcription in the generation of DNA damage.
Yes, that's fair enough. That's a more sophisticated way of building what we know already, fine-tuning the defense mechanisms so that they work better, and that's important enough.
So going back also to the one thing that I'm curious about is that we talked about aging, and one thing that can improve aging is fasting or caloric restriction. And this has been proven in many different organisms to really expand lifespan. And also, so my question to you is, do you think that this has something to do with generation of DNA damage?
Only to a small extent. My understanding is that protective effect you can see from reducing your caloric intake. It's only noticeable when you are sort of semi-starved.
Yes.
So perhaps you could prolong your life a little bit by being hungry all the time.
Yes, yes.
But that's unpleasant.
It is, yes.
So it becomes an individual choice. I think I would prefer to continue eating when I'm hungry and accept that from what we know today, that might have a small effect with effect of longer life.
We have talked about the spontaneous decay of DNA. DNA is, of course, carrying the genetic material. This is critically important. But also, there is damage to other macromolecules. Of course, RNA, but then also proteins and membranes. And when it comes to aging, what do you think is the contribution of the damage to the other macromolecules rather than to the DNA?
That's a question that's debated very much right now. I would put my own money on DNA because the genetic information is, after all, what everything else is expressed from. But my colleague Miro Radman
in Croatia,
Croatia is now, he has come up with some pretty striking arguments why some forms of protein damage might also be important.
Of course, yes.
So I think the big problem is DNA damage is not so easy. You can't replace the DNA. You have to, well, you can cut out a section of it and replace that section, and that's the DNA repair that we have both been working on. Protein repair doesn't really occur. If you destroy a protein, you can make a new one, but to replace the damaged one. But you don't take a damaged protein molecule and repair it.
But you also have protecting the proteins, which Radman is talking about.
Yes.
That it's important to protect the proteins, to keep them intact, in order to be able to repair, for instance, DNA.
Yep.
So let me switch a little bit and talk about cancer. And so DNA repair and damage is very important in cancer. And over the last few years, we have had a lot of sequence information from a lot of cancers, enabling us to study the mutational signatures, which can give a hint on the damage is occurring. And this is coming from Mike Stratton and others' lab, and I'm pleased to say that we have Ludmila Alexandrov coming here to SciLife Lab to receive a prize. So he received a prize, the magazine Science and Science SciLife Lab prize for young investigators.
Oh, that's very good.
So so he's getting this prize the day before you, on the 9th of December.
Okay.
So this is also going along the lines, you know,
Yes,
you were correct.
I've been to Cambridge to discuss these problems with Mike Stratton, who is an excellent scientist, and I think they are doing some fascinating work on this.
So, to my mind, you know, this is really demonstrating that the deamination is very important.
Yep.
But also, I'm very surprised to see so little oxygen damage causing mutations in cancer. I would anticipate that oxygen would be fairly important. We consume a lot of oxygen. We have water molecules around.
I'm not surprised by that, because in the cell nucleus, and there are some excellent old data on this that have not been paid attention to enough. There's actually very little active oxygen in the cell nucleus. And this is not by chance. Our cells are constructed so that we have the oxygen metabolism that's required in a separate little organelle called mitochondria. That's where all the oxygen business is handled.
So you keep the oxygen, the damaging oxygen, away from the DNA.
Exactly. So in the nucleus, there's very little active oxygen.
And this is also the reason why, you know, the oxygen repair mechanisms, such as OGG1, the glycosylase, is not essential. Or do you think it's the redundance between them, or?
I think both, but when it comes to DNA damage, there are other agents in the cell that I think are more important than active oxygen. Water is very dangerous, but there's nothing we can do about that. But with regard to oxygen, I think there are other small metabolites in the cell that are more important. We have been interested in a small coenzyme called S-adenosylmethionine, because that is the methyl donor in cells. That's a coin you use in transmethylation reactions. But there are other small molecules. Formaldehyde is formed, it's a very reactive substance, and is formed as part of metabolism. And there are other things we know. If you take a classical biochemical textbook and you go through page by page what happens, there are several small reactive molecules formed in cells. Do they damage the DNA? We don't know. Nobody has looked yet. And if they do, there will have to be separate repair enzymes to deal with them.
So do you think there's still a lot of new pathways and players to be identified in the DNA repair field?
Yes, I think so.
And indeed, we have found a lot of players involved in chromatin organization being critically important for genome maintenance. But there can also be some direct DNA repair enzymes that are important. I heard also that you were inspired by Walter Bodmer in your early days. And can you tell me about, I've been working very close to Bodmer also in Oxford, and had the opportunity to talk a lot of long hours with him. And but can you please tell me what inspired you from him?
Well, Walter Bodmer had become the new director of the biggest cancer research organization in Britain, which at that time was called Imperial Cancer Research Fund. And he offered me a job, which was a very attractive job, to set up a whole new research laboratory. Not a big one, but 10 research groups or so. And we would have very free hand with what we could do, concentrate on various aspects of how cells respond to DNA damage and so on.
So it's also from Walter, and there's been this argument, you know, the mutator hypothesis, that you have cancers are generating more mutations themselves, which Larry Loeb has founded, and then it's been Walter Bodmer's view that it has been more of an evolution path of cancer development, that you
Well, it's an ongoing argument, but in this particular case, I probably side up with Laureate, that it's not just a selection process. There are things happening that are very interesting and that Loeb and others are trying to characterize, and perhaps you are trying to characterize too.
Yeah, and I think I side a little bit with Walter that the human evolution is so important, and we see the heterogeneity being critical.
You know, the answer will be that everybody is right.
Yes,
there is
of
an
course,
evolutionary aspect,
yes,
but there is also a selection process.
Of course. So I would like to also discuss a little bit about, you know, how can we exploit the mutations in cancer with treatments. And we have seen we have inherited mutations that have passed through generations, increasing risk of cancer, such as the mutations in mismatch repair, giving colorectal cancer, and mutations in homologous recombination, giving rise to breast and ovarian cancer. We know now, when sequencing all of these cancers, that a vast majority of these cancers have DNA repair defects. So, in your words, how do you think that we best exploit this?
Well, here is a good topic for people who are interested in bioinformatics, because with the new DNA sequencing methods, we are drowning in interesting information. The naive question was, will we find a mutation in a cancer cell? Well, the problem is we find 100 mutations in the cancer cell, and some of them is just background noise, just something that happens in our cell because the DNA isn't as stable as was believed previously. But then there are occasional dangerous ones, and that's by the techniques of or molecular approach of epidemiology. One can hope to identify those against the background noise. But up to now, there's been so much background noise that people seem a little bewildered by how can we find the really important changes.
Yes.
But the question of a few years ago, are the genetic changes in cancer cells? Everybody agrees, yes. There are too many of them, which are the really important ones, because if they were all important ones, then well, it would be very difficult to have organized life. So we can also agree on that most of the mutations we see in cancer cells are just noise. It also happens in our own cells that are not malignant.
So we and others have shown that you can target a backup pathway in cancer cells that have lost the first repair pathway.
Yes.
And the PARP inhibitors are now approved and working, the DNA repair inhibitors in cancer. Yep. Do you think that this is a good strategy also for the future? Is there more to find in this area?
I think it's an excellent strategy, and you are just on the beginning of the exciting developments here. It seems fairly common that cancer cells, because they grow so fast and in a sloppy way, that occasionally things go wrong and they lose a pathway. And then, as you see, the cell is ready for this because there's a backup pathway. But now it means there's, if you want to knock out that cancer cell, instead of having two targets, which are difficult to knock out at the same time, there's only one target in those cells, and you have to identify that target. And then there's good hope that you can really slow down the cancer growth.
We also talked about that cancer in general has a different biochemistry as compared to normal cells. They switch to anaerobic glycolysis, and this is also has implications for type of lesions are formed in DNA.
Yes, that is
and
right.
Probably also implications to what repair proteins are required for the different cell types to survive. Yeah. And so if we can find what do you see? Do you think that there is an opportunity to find new enzymes that we can target, DNA repair enzymes?
Yeah. We all have to go back to biochemistry or metabolism and understand much better what's going on. It was considered for a long time a bit old-fashioned and not so exciting, but now I think it's a very core of no cancer research.
So if you were young today and you want to find a treatment for cancer, what would be your advice to the young scientists?
Study biochemistry and metabolism and find out how cells work. If you want to understand how cancer arises, you first have to understand in better detail than we have today, how does a normal cell work? How does it interact with other cells? It's there something can go wrong, and that can result in a cancer or premature aging and all kinds of other problems.
So now what you're saying now is basically going back to Otto Warburg, who said that the resultant, the cure for cancer is within the biochemistry of the normal cells and cancer cells, the difference.
There is an important lesson here, because Warburg said these visionary things over 50 years ago. At the same time, he said some incredibly silly things which we know in an old-fashioned and can't possibly be true. And some people then assume that everything he said was wrong, but in fact, most of it he said was right, and it was just amazing.
Yes. And I think this is also a lesson, You should be firm in your beliefs and not in beliefs, but you should look at the science and look at the details, which you had attention to detail, to what you observed. And once you observe things, then those observations are true. And believing in observations that are true is very important in science. And then, of course, you can offer alternative explanations. But to stick with the science and stick with attention to detail. And as I say, to people in my lab and in other labs, that your future is looking down the microscope to attention to detail and see something different, see something unique that you think is difficult to explain, and then study it more thoroughly.
Yeah. The sad thing is that some people who have some biased ideas, they might see something that's actually very important, and they ignore it because it doesn't agree with what they believed would happen. So it's very important to have an open mind.
Yes, you have to challenge your own convictions, and think that you need to be open-minded, and I think that is the key. And probably this is something we could end with. We should be open-minded. We should also focus on the really important, the core biochemistry of the cell, and really understand the difference and between the normal and cancer, and also understand the normal core of the biochemistry of the cell, just like you did. And there's still a lot of missing links there.
I think it's also important to remember that clearly I have done some useful things, otherwise I wouldn't have got this Nobel Prize. But it might well be that some of the things I've said are completely wrong.
Yes, well,
and
time
then
will
somebody
tell,
has to challenge it.
But I think you have ultimately proven, me for one, and among me, tens of thousands of scientists have proved you right.
Yeah.
So I think it's going to be difficult to challenge your original paradigms.
That I don't think can be done, because it has now been repeated by so many other scientists. And that's in the essence of science. Your results should be reproducible.
Yes, so with that I would like to again congratulate you, Thomas, to the fantastic award, and for thanking you for coming here and taking some time visiting us here at SciLife Lab at Karolinska.
Pleasure to be here.
Thank you.
Thank you, and good luck.